Good morning. I want to share with you today some new technologies that we're developing for the analysis of monoclonal antibodies. As we go through it, you'll understand what these terms mean. So one of the things we're going to look at is what we call high-throughput molecular recognition as a way to analyze monoclonal antibodies. So it's an alternative actually to the way people currently analyze monoclonal antibodies and proteins to determine their quality and their biological efficacy. So the FDA has responsibility in North America, in the US, in assessing how people should determine the quality of their products that are therapeutic proteins. And so the FDA then had the idea a long time ago that you design a therapeutic product to have a certain structure, to behave in a certain way. And they call it quality by design. And if you have a quality by design, then you have certain critical features that will be in that molecule, and they refer to those as critical quality attributes. And if you look at the critical quality attributes as you come down, they do it in a CPA, a so-called performance attribute, is in fact whether the way that the molecule actually acts. So the function of therapeutic protein is to be therapeutic, and that's what a CPA is. And if you look at that, then the FDA thinks one of the ways that you should determine that is by the critical structure attributes are there, that you have to go in and demonstrate that you have the right critical structure attributes. And these are what you would show then that is there. For example, oxidation, deamidation, various things that can be modified in the molecule, and look at it. And these are then the methods by which you do that, and it's done primarily today by mass spectrometry. And as I said, the way you do that then is to take the molecule apart and look to see if it's been oxidized and all of its deamidation and so forth. And that's one way to do it, by basically taking the molecule apart and looking at all the molecular features. But if you look at that, you look at all these attributes you can look at, that takes a lot of time, to do it. And if you think about so many things that we do in manufacturing things, that you want to make decisions on whether the process is working, and you can go ahead with it, based on knowing very quickly whether there's a mistake in the molecule, and that is time-dependent data decision-making. Our alternative then is to look at whether the molecule actually works, and whether the molecule actually works as an early warning in the manufacturing process or in the development process that the molecule will not actually be biologically active because it doesn't test active. And so we have an activity assay. And so our approach then is to develop early warning systems that assess the quality of molecules, and we do it by recognizing the features of it, whether the features of it on a molecular basis are correct. And so if you look at that, one of the things that happened in recent years after 2000 that we didn't know before 2000, and the FDA didn't either, is that molecules are much more complicated than we think. And this is genetically why that is so. This looks really complicated. And what this really complicated thing shows you is that as a molecule is being made from a particular gene, that it can be made in many different ways. In the human body, we have about 23,000 genes. And the surprising thing is we make over 100,000 or 1 million proteins from that 23,000 genes, and that surprised the world. In 2000, we finally figured out how that's so. And a gene can be expressed in many ways and make a family of molecules from a single gene. And it turns out, as we see over here, that you can actually get 50 to 100 different forms of a molecule, all that have different biological activities, but they come from a single gene. And so what that means is as you use a mammalian cell to manufacture a monoclonal antibody, that monoclonal antibody is not one type of molecule. It's many types of molecules. Some of them will be biologically active and some not. And so that's the whole issue then that you have, is to determine what percentage of all the molecules that are made that are in fact so-called monoclonal antibodies would actually be biologically active, would be therapeutically effective. So instead of taking the molecule apart to see how it's made, we do it another way. And we do it by molecular recognition. And what we try to do is go in and find some molecule, some coding agent, if you will, that will only see the molecule that we want to see, that will only see in the thousands and thousands of molecules that are made in a biological reactor. We only want to see, and you only want to know, the small number that are monoclonal antibodies that are biologically active. And so what we do is we take a little coding agent, we call it a molecular recognition agent that is coded. And its coding is it has a fluorescent tag on it, and it has another tag on it that recognizes the molecular feature. So now if we take monoclonal antibodies, and there are actually multiple forms of them, there's one that is just one antibody itself, and then some of them stick together in dimers, and some stick together in trimers, and so forth, that if we throw this reagent in, it will recognize that it's an antibody and bind to it. And when it binds to it, the code it puts on it is it makes it fluorescent. It makes it have an optical feature that's only present on that molecule. There can be 1,000 to 10,000 other proteins in that mixture, but the only one that will light up this way is the monoclonal antibody. So, how do you do all this, or where do you do all this? How do you do this work? Well, currently it's done in a laboratory that by anywhere from 5 to 6 to 10 people, and it's like the old expression, it takes a village to raise a child. Well, in the way that monoclonal antibodies are done today, it takes a village to analyze the antibody. And so we circumvent all that by doing it in a simple little column like this. And we say everything happens here. And what I'll do with the rest of the talk is tell you how we can do all of that in that one little tube. And that little column does the following. And when we start is to take a sample out of a fermenter, and this is that sample, and we put it in a tube, and it's going to move through this tube at a certain velocity. And we then take that coating agent that you saw before, and we put that coating agent in too, but that coating agent is going through the column continuously. So the whole column, this whole tube, is filled with the coating agent. And it moves very slowly through the column, but the antibodies, because they're bigger, will move fast. And as a consequence, these molecules will go past these molecules. As they go past them, what will happen is the little coating agent then will stick to the antibody and cause it to light up. Now that normally would be done outside the column in the normal way in a test tube. And you'd have to do that manually and shake it up and so forth. But the reason we don't have to shake it up is because the difference in velocity causes this big molecule to go past this little one. And when it does, you get a reaction. Now after you get this reaction, now the molecules went from looking like this to looking like this. And that all happens here in the first part of the column. And after it happens now, we're interested in seeing how much of this, this, and this you have. And so as it goes down the column then separates these molecules from each other in separate zones as they go through, and they emerge from the back end of this column at different times. And the big molecule, because we're separating them by size, the big molecule is first, and then is followed by the smaller molecule, and finally by the smallest molecule. This particular thing right here, this particular negative peak, is what happened down here. And as this reagent was consumed and stuck to here, it causes this negative peak. So it's the fluorescent coating zone. But then as it separates, this molecule here is smaller, so it comes out there. But this molecule, being the biggest one, travels fastest of all, and it comes out first. So that's how we use this molecular recognition thing right here to be able to separate molecules over here that you can then quantify and know the relative amount. Now, why do you want to know this? Well, this is the therapeutic form. This form over here that's formed accidentally can be toxic, Or it can cause, if you put it in a patient, it can cause the production of antibodies against it. So what's happening then is your body would produce antibodies that attack the therapeutic agent, and you don't want that. So that's why you want to know when you manufacture a monoclonal antibody whether you're doing this or not. And as we can see here, with this particular form, this particular sample, that it has a very small amount of dimer. And it's small enough that it's probably not a big problem. It's about 1 or 2% of the total product is dimer. And then you see over here, this one, there's almost none of it. So that's what goes on in the column, and that's what we mean by a molecular recognition analysis. Okay, and so we blow that up a little bit more. And now what we look at is, can we actually determine how much of that substance is being made? And yes, indeed, you can. And as you go up, you see as the increasing amount, and then the linear dynamic range shows that, in fact, is a straight line. And you also see the negative peak here, that's the coating agent that's sucked up. And at high concentration, it takes more of it, so it takes a little bit longer to do that. But you don't get any band spreading because the speed with which the big molecule goes through is not affected by the binding of the little molecule. And this is a blow up here that you can see the dimer here. There are multiple forms of it that increases, and you can't see any tetramer at all. So what's missing? What don't you see? Well, that sample we put in the column actually has between 1,000 and 2,000 other proteins in it, but you don't see any of them. And you don't see any of them because of coding. The coding agent only goes on monoclonal antibodies. The coding agent does not go on the other 1,000 to 2,000 proteins, so you don't see them. And that's why there's nothing else on the chromatogram. You see a little tiny peak out in here, a little tiny peak that may be due to some of the coding agent binding to other proteins, but it's so small that you don't actually see it, it doesn't interfere. So going on, if you actually take this and go out and look at the production of a monoclonal antibody in a fermenter, that occurs in about 2 weeks. And what you see is the production of monoclonal antibody with our molecular recognition assay. And this is two different production runs. And you see how similar they are. They're not identical, but you wouldn't expect them to be identical. And this is an alternative method to do it in the red. So the blue is with our molecular recognition assay. The red is with another way. And the other way is that you take and get rid of all the other proteins that would interfere. And the way you do that is to use affinity chromatography, and the affinity chromatography column is a protein A column. Protein A is a protein that binds to antibodies, and then you release the antibodies from the protein A column and run it on a size exclusion chromatography column. And what you see is it gets somewhat similar data, but it takes much longer to do it, and you see the reproducibility is very poor, and it doesn't necessarily give you the same answer. And we went off and looked at with alternative means in another way, looked at concentration, and at the molecular recognition reagents that I'll show you in a minute, very, very little in run-to-run. We also went off and looked at this with multiple antibodies, and you can see an interesting thing then. You can see that the method is somewhat universal, because these antibodies over here are different from each other, but yet the chromatograms look almost identical, although they're different antibodies. And so what we designed our reagent to do is only see antibodies, and it will see all antibodies in almost the same way, almost identical. So the nice thing about that then is we designed the assay that you can turn around and assay large numbers of different antibodies with the same test system. So this is if you go off and run a large number of tests that you can see. This is, for example, 600 runs, and that would be almost during the entire 2 weeks of a production run, the degree of variation you get, and this is the ratio of the the high molecular weight dimer, trimer, tetramer assays, which you can be toxic and so forth, you don't want to have, versus the good form. And you can see the degree of variation then is pretty tight. We go on and look at that. Here I told you that you can assay different forms of antibodies, and it turns out in monoclonal antibodies they're called immunoglobulin G types, but there's type 1, 2, and 4. And it shows that this assay, in fact, works almost identically with all of them. As you go down in concentration, to get smaller and smaller concentrations, of course, the variation in the assay increases. But you can see that the percentage of this dimer that you have is very small out of the total. It's 1/10%, 0.2%. And as you go to lower and lower concentration, you would expect more variation in the assay. But that's pretty tight. That's very little variation. So in some assays, you want to go very quickly. And for example, in clone selection and things like that, you want to do a large number of assays, maybe 1,000 assays a day. So this is what a normal chromatogram would look like, as we showed you. But if you look at that chromatogram, you see that the last part of it, there's very little there. There's nothing there that you're really interested in. So could you run the assays faster? And yes, you can, because as you see here, we're able to make 4 or 5 injections before any of these negative peaks come out. And so if we use a chromatography column here, one column that does 4 or 5 injections, and then we switch it offline and switch a new column in, and while the new column is running another set, we throw away the 5 from here, throw it to waste. Now you can get this sort of a thing here, where you're doing a sample every 1.4 minutes, or every 84 seconds, you're doing another sample. And what that says is you can do 40 samples an hour, or 1,000 samples a day. And there is a need to do that in clone selection and things like that, where you want to look at thousands and thousands of things over the course of a week or two to go in and choose the best cell line to produce your antibody. So one of the things we want to talk about here also is the significance of the negative peak. What does that negative peak mean? The negative peak, remember I told you, is as the big molecule goes by the little coating agent, it sucks it up. And as it sucks it up, it decreases its concentration, and that's what the negative peak is. So it shows up over here as a positive peak. But one of the things that's also very significant that I'll show here is that if you look at the area of the negative peak versus the area of the positive peak, you get more fluorescence from the positive peak than you do the negative peak. And what that means, that's very significant, is when this coating agent binds to the monoclonal antibody, its fluorescence increases. So there's an amplification of the fluorescence, and that amplification of the fluorescence increases the sensitivity. And so that has another meaning also, is that you can run things continuously and don't have to do the separation, just get the amplification of fluorescence. So anytime that you have a test tube and we put that coating agent in a test tube that has antibody in it, the fluorescence will increase. And that is terribly valuable, because now it says that in a test tube that has 2,000 or 3,000 proteins, one of which is your antibody, if you throw in the coating reagent, the fluorescence will increase, and that total increase in fluorescence will be just due to your antibody, nothing else. You don't even have to do a separation. And so there are multiple ways that you can use this system. One of the ways is that you can use it to do 2 or 3 kinds of assays. One type of assay you can do where you get the discrimination between dimers and monomers and so forth here. Or the other way is you can say, I don't really care about that. I just want to know how much antibody I get. So if instead of going through the chromatography column, you turn around and you add the reagents here and just go through a mixer, then you get this assay where you see all of them come out at once. And that we refer to as a rapid titer assay because it tells you how much antibody you're making, not necessarily if you have any dimer, trimer, tetramer, and so forth. And that also is valuable. And so it means you can go back and forth between these two in a course of 1 or 2 minutes or so and do multiple kinds of things in one system where you can't do that any other way. Still another way that you can use the system is instead of making the derivatives and doing all the fluorescence, everything inside the column, you can turn it around and do it another way. You can run things through the chromatography column, and because you get amplification, you mix the fluorophore in at the back of the column, do the molecular recognition at the back of the column over here, and see the increase in fluorescence, but you're still doing a separation here and only looking at the peaks that are coming out of the column. So there are 2 or 3 ways to use this technology, but the actual mechanism by which it works is the same. So there's yet another question that you have. That an antibody is designed, and the way antibodies work is that they have a portion of the antibody out here that binds to a molecule in the biological system that it's supposed to use to treat a disease. And that part of the molecule is called paratope. And one of the things that determines the quality or efficacy of an antibody is whether the paratope is functional. And it may or may not be. So one of the things you're concerned about then is to be able to make an analysis of whether the paratope is present in the molecule. And I take over here a case that you can have. An antibody has 2 active regions that bind to other molecules and that you would use as a biological function. And so the paratopes would be here, the part that it binds to some of the molecule. The little yellow dot up here says that this antibody, it has 2 identical parts, but there's one part that's defective. Or there's another thing you can imagine is that they're both parts that are defective. And how can you know that about a molecule that your antibody is or is not defective? And the way that it's done, and the way that you can find that out is to actually take the antibody and give it to a patient and see if it's effective, see if it works. Or you might be able to dream up some sort of an assay to do on a test tube that would do that. But what you want is to find out, to try to develop a mimic of what would happen in the body if you injected the monoclonal antibody. And the way we do it is in the following way. We take the biological molecule that's inside the cell and take that molecule and see if the antibody will bind to it. But remember, it's in the presence of, the antibody is in the presence of thousands of other proteins. So now what we're asking it to do is do more than just recognize the antibody and whether it's present with 1,000 or 2,000 other molecules. We want to know also, is it biologically active? So 2 things, and we up the ante on what we have to be able to do. So now, if we take the molecule that it's supposed to bind to and make that fluorescent, like we made this guy fluorescent, but we put a different fluorophore on it, now when we look at it, we see 2 pieces of fluorescence. One is we see the fluorescence of this, so we know it's a monoclonal antibody. And we see it's the fluorescence of this, so we know that it bound to the antibody. And remember, we can do yet another thing. We can separate these molecules. And because this has size, now what we see is that this molecule, which will bind 2 of these, is over here. This one, which will bind 1, is here. And you can differentiate between all of these. You can differentiate between all of these forms. And it turns out the really incredible thing is that you can do this in 3 or 4 minutes. So you can know in 3 or 4 minutes whether the antibody is actually functional. And why is that important? Well, we learned in the COVID vaccine era that the public was very concerned about whether the molecule that they were injecting into them was efficacious. Did it work? Was it any good? And this kind of an assay can determine that in 3 or 4 minutes. And so what it means is in manufacturing and all kinds of things, you can do very quick things that give you an early warning that a molecule is active, it's not active, and in fact, if it's not active, the percentage of the whole thing that does or does not work. Now going to the conclusions, that this kind of molecular recognition assay can be done in 10 minutes. And we can find out all kinds of features about the molecule, but the most important one in molecular recognition was whether the molecule works or not. We know whether it's biologically active, as opposed to just doing an analysis where you find it has the right features or not. That's not the question. The question is a therapeutic protein therapeutic, and we can know that in less than 10 minutes. You also saw that with the little tube that you can hold in one hand, that these molecular recognition assays are inherently simple. Very, very simple. It doesn't take any big fancy instrument to do that. And the other thing you saw is that you can determine 2 or 3 things about the molecule simultaneously within the 2 or 3 minutes. And we're assessing biological activity. And we're doing it in the intact molecule. We don't have to take it apart. And that's why that you can do these kinds of things very quickly. And we call these Luminon assays. And that these Luminon assays, the thing that's so good about them and so great is you can use them in production while you're actually producing the antibody. You can use them while you're purifying it. You can use them in process development as you're discovering the antibody. And finally, you can use them in formulation. And interestingly enough, not generally recognized, is that even when you're putting a therapeutic agent in the bottle, then it goes to the patient, that you put them in at a high concentration and so forth. That can change the efficacy of the molecule, so you even need to assay efficacy and safety at that point. And that's why a very simple assay is very important. And what we look at these assays as doing as being an early warning method, an early warning that there's something wrong. And in the early warnings, such as with COVID vaccine, there was a lot of COVID vaccine manufactured that they didn't realize until just before they shipped it that there was something wrong with it. And what had happened is that in the formulation, they made a mistake and mixed 2 things together that shouldn't be. And so that's why the early warnings, very rapid analyses in looking at therapeutic proteins is a big deal. And after you do the very early warning, you can go back then and do in-depth analyses in other ways, like by mass spectrometry or so forth, and find out why the molecule didn't work. And is that something that we predicted, or is this an entirely unknown thing? And for example, what this does, it's very interesting, is that there are sometimes mistakes that happen that are not predictable. In other words, you don't know what you don't know. And so you can do unpredictable things like people making a physical mistake in the order in which they do things and so forth. So it's very important that you have a test that shows you that the unexpected happened. And the unexpected is that the molecule doesn't work. And so that's why it's so important that you have tests that determine whether a molecule works or not. Thank you.